Oh, my dear students, today I will be dealing with the dihybrid cross done by Mendel. On the basis of this dihybrid cross, Mendel formulated the second law, that is independent of assortment. So first, before going to the second law of Mendel, we shall try to understand what is the meaning of dihybrid cross and how it is done and what are the different aspects selected for the dihybrid cross. Let us see in detail. So first of all, we have to recall the first class. In that first class, we have seen monohybrid cross. So what is monohybrid cross? A cross which is conducted for single character, single pair of contrasting characters. In the beginning, we have seen that Mendel took a tall plant and a dwarf plant and crossed, and he got the results that we have seen already, and that is called as the monohybrid cross. So now he is conducting the dihybrid cross. Dihybrid cross states that the dihybrid cross is the one where two contrasting characters are taken at a time. Two characters are taken at a time. So because of this, it is called a dihybrid. Di means two. So two characters are taken here, and these two characters are pertaining to the characters present in the garden piece. Okay, the dihybrid cross. Dihybrid cross. So in this dihybrid cross, Mendel selected a garden pea. So here he selects the shape of the seed and the color of the seed coat. Shape of the seed, there are two shapes available. We have seen in the first class, 7 contrasting characters were selected by Mendel, and out of it the first character is height, and height he has selected for the monohybrid cross. And for the dihybrid cross, out of 7, he selects 2 characters. One is the shape of the seed, and second one is the color of the seed coat. Shape of the seed means what? So in shape, we have seen 2 contrasting characters. One is round seed and another one is wrinkled seed. But here again, round is dominant over wrinkled. Round is dominant over wrinkled. And another character selected by Mendel for this dihybrid cross was the color of the seed coat. Okay, so color of the seed coat, again 2 colors, green and yellow. Green and yellow. Yellow and green you can call. Green, yellow and green. But here also, yellow is dominant over green color. So such a thing he has taken. I will just write here. So first he selected round yellow seed. That means the plant selected for dihybrid cross was producing the round seeds, which are round, and the color of the seed coat was yellow. You must have seen yellow peas, haldi batani anta kariteve. So round yellow, and he crossed with another one, that is wrinkled green. He takes, and here the seeds are wrinkled. Say for example, the seeds are like this, but here the seeds are round. So such thing he took and he conducted the cross. But here he gives, this is the phenotype of the plant, and he is now taking the genotype. Genotype of the parent was, so each character is controlled by a pair of factors. So round is denoted or controlled by a pair of genes, what are known as capital R, because I have told you already that round is dominant over wrinkled, so it has to be represented by capital letters. So that is capital R. Yellow is dominant over green, so it is capital Y. And correspondingly, the what you call the recessive character should be written by means of the corresponding small letters, that is small r, small y, small y. We have to take this one. Small y and small y. We have to consider. And the gametes, so this we shall call it as parental generation 1, and it is producing the gametes now. Gametes are capital R, capital Y, only because similar homozygous are there, only one gamete can be taken, and small r, small y. Two gametes are produced, and because of the hybridization, fertilization, a plant is produced, capital R, capital Y, small y, and this we call as F1 plant. F1 plant, first filial generation. So F1 plant is produced here, and what is the phenotype of this one? We got the genotype. What is the phenotype of this? Capital R, capital Y, small y. That means capital R always produces the round shaped seeds. Capital Y is producing the yellow colored seeds, yellow colored seed coat. So in presence of capital R, small r has no expression because of recessive nature. In presence of capital Y, the small y has no expression because of the recessive. So this plant must be round and yellow. Round yellow. That means the offspring produced in the F1 generation was having the character round shaped seed, and the seed coat was yellow, and ultimately this must be called as hybrid, because this is the plant produced in the F1 generation due to the cross between the two different varieties, two different contrasting characters. So this is called as hybrid. Hybrid is the plant obtained in the F1 generation. Okay. Now after this, Mendel was not satisfied, and he wanted to know where this recessive characters gone. So because of that, he was very much interested to know the next generation, that is he called parental generation 2. Now he is selfing the F1 plants. F1 plants are self here, that is round yellow crossed with F1 round yellow we shall call, and F1 round yellow. Okay, so capital R, small r, capital Y, small y, capital R, capital Y, small y is the genotype of the F1 hybrid, we have seen already. And the gametes produced by the F1 plant, unlike the parental plant, the F1 plant will produce 4 gametes because of the heterozygous nature. So capital R, capital Y, first gamete, then capital R, small y, then small r, capital Y, and small r, small y. So 4 gametes are produced here. Likewise, since the genotype of both the plant is same type of gametes, capital R, capital Y, then capital R, small y, small r, capital Y, small r, small y. So 4 gametes are produced now. So after when the 4 gametes are produced here, so the Mendel has to put the checkerboard now. So what is the shape of the checkerboard now? What is the size? So we have to, depending upon the number of gametes, we have to decide the size of the number of columns and rows in the checkerboard. So since there are 4 gametes here, 4 gametes here, so the checkerboard must be 4 by 4. 4 this side, 4 this side. Totally we are getting 16 rows and 16 columns. So 16 total columns are available now. So we have to write. So this is the checkerboard. 1, 2, 3, 4. Checkerboard here. So 1, 2, 3, 4. So this is the checkerboard, and in this one we have to write. So on this corner, as usual, male gametes this side, female gametes this side. So we have to write 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This is the method of preparing or writing the checkerboard. Checkerboard is like this now. Now we have to write the gametes here. Gametes, first one is capital R, capital Y. First gamete, capital R, small y, small r, capital Y, small r, small y. This side also same gametes. Capital R, capital R, capital Y. Sorry. So here capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So 4 gametes this side, 4 gametes this side. And as a rule of the checkerboard, you have to take this gamete and multiply and write here. So the first one is capital R, capital R, capital Y, capital Y. Second one, capital R, capital R, capital Y, small y. And again, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. And capital R, capital R, capital Y, small y, capital R, then small y, and here capital R, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, capital Y, small r, capital Y, small y, and here capital R, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, small y, small y. Like this, 16 plants are available now. After crossing or after selfing the F1 plant, 16 total plants are available, and these 16 plants are available, and here in this checkerboard we know only the genotypes. So we have to convert these genotypes into phenotypes now. So we have to write the phenotypes here. Phenotypes. So here number 1. What is the first plant available here? First plant in the first square of the checkerboard, we are observing capital R, capital Y, capital Y. So we have to remember one important thing. If only one capital R is present, of course here 2 capital R are present, but at least if one capital R is present, so that must be round only. Okay, and if there are 2 round, it must be round, and if there is one capital Y, it must be yellow. If the 2 are present, well and good. If there is absence of capital Y, so then it must be another color, that is green color. So okay, now come to here, capital R, capital Y, it must be round yellow. Round yellow, first plant. Now we have to note down how many round yellow plants are available in this checkerboard. So now write the number. First plant is round yellow. Second plant is capital R, capital Y. So that means second plant is also round yellow. Capital R is present, capital Y is present. Third is also round yellow. Capital R is present, capital Y is present. Fourth is also round yellow. Fifth round yellow. Both are present here. Capital R is present, capital Y is present. But sixth plant you have to observe. Capital R is present, so it must be round. But capital Y is absent here. It must not be round yellow. It must be some other. We shall not take into account now. Seventh plant again round yellow because of capital R, capital Y. Eighth plant, it is not round yellow because round is present but capital Y is absent. And ninth plant is again the round yellow. 10th plant is also round yellow. because of the presence of capital R and capital Y both. But here capital R is absent, only small r present. Capital Y are present, but it must not be round yellow. The 12th plant, it is capital Y is present, but capital R is absent. It is not round yellow. Coming to 13th plant, so that is capital R, capital Y, both are present, so it must be round yellow. So further, there is no capital R, capital Y, so it must be totally how many? 1, 2, 3, 4, 5, 6, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9 plants are available now. Totally 9. What are the 9 plants here? 9 plants are round yellow. It is called as parental generation. Parental type of plants are available. Okay, now second we have to observe. What is the second one? Second one means which number is missing here? 1, 2, 3, 4, 5, 6, 7 is missing. So what is the 7th plant here? 6 is, okay, 6 is also round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 10. 9, 10, 13. Okay. 1, 2, 3, 4, 5, 7, 7, 9, 10. Okay, 7, 9, 7 is there. Okay. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9 plants are available. Now, we have to observe which number is missing here. 6 is missing. What is the 6th plant? 6th plant is capital R present, so it must be round. Capital Y is not there, so only small y is there. So it must be a new variety available here. That is round green. Round green. So round green is 6th plant. Then round green 8th plant. Okay. Then round green is 14th. 14 plant available. Totally 3 are available now. And third one, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 is missing here. So 11 you can observe small r, capital Y. So capital R is absent. It must be wrinkled, and capital Y is present. It must be green, yellow, wrinkled yellow. Wrinkled yellow. Wrinkled yellow. It must be 11th, and after 11th, wrinkled yellow, 12th. 12th is also there. Okay, and 15th is also there. So totally there are 3 plants available. Round green, wrinkled yellow. So they are available 3 each. And the last one is only 1 plant is available here. That is wrinkled green. So that is 16th plant. So it must be 1. So this is called as dihybrid cross. Dihybrid cross is nothing but it is the cross conducted by Mendel by selecting 2 characters at a time. Each character is having 2 contrasting characters, round yellow and wrinkled green. When he has crossed like this, he got the ratio 9:3:3:1. So this is called as dihybrid cross ratio. Dihybrid cross ratio. Okay, so on the basis of this, the total plants available here are 16, and out of 16, 9 plants are it's called as parental type, and 9 plants are resembling the dominant parent, and 1 plant was resembling the recessive parent, and 6 plants are new varieties. So they are called as the new varieties available which were not present. So that is called as recombinant types available. So this is called as the dihybrid cross ratio. Now on the basis of this, Mendel formulated the second law. So first law we have seen already. When a pair of factors are brought together in a hybrid, they segregate or they separate during the formation of gametes. This is called as the first law of Mendel. But the second law of Mendel, second law of Mendel is called as law of independent assortment. It is called as law of independent assortment. So how to understand this law of independent assortment? We have to see now how we have to take into account. How to understand law of independent assortment now. So again go back for the crossing of the plants. So Mendel took capital R, capital Y, cross with small r, small y. So during the formation of gametes, capital R, capital Y came in the first gamete from the dominant parent. Small r and small y came into another gamete from the recessive parent. But when these two are fertilized, so capital R, capital Y, small y came into the F1 hybrid. This came in the F1 hybrid. Here the Mendel is interested on the basis of the behavior of the gametes, behavior of the genes during the entry of gametes. He formulated the second law, what is known as law of independent assortment. How? So during the formation of gametes, you can see here this R need not be always present with small r. During the formation of gametes, this R may go and join with the capital Y, and this R may go and join with the assort with the small y. This small r may go and assort with capital Y, this may go and assort with this one. So each gene or the factor, in the language of Mendel we have to call it as factor. Each factor is independent to join with any of the factors. So capital R is assorting with capital Y. Same capital R may go and assort with small y. So this, because of this, we are taking here capital R, capital Y. So you can see here capital R and capital Y came in one gamete. Likewise, capital R and small y also came in another gamete. See the assortment now. And same way, small r is assorting with capital Y and small r is assorting with small y. So this type of thing is called as independent assortment. Nobody can restrict the genes for assorting with a particular gene. You have to go with this only. You have to go with this one. Nobody can restrict. So they have got 100% freedom to go and assort with any of the factors during the formation of gametes. So this is called as the Mendel's second law, the law of independent assortment. So with this, the dihybrid cross and law of independent assortment is completed now. Again, the problem came that when the plant, F1 plant was available, what was the phenotype of the F1 plant? It is round yellow. But according to Mendel, it is round yellow, but it is heterozygous in nature. But the parental round yellow plant is homozygous. So internally they are different, externally they are same. Morphologically they are same. Internally they are different. One is totally homozygous, another one is totally heterozygous, and again the objection came. How do you say that it is heterozygous and how do you say that it is homozygous? Because externally you cannot observe the genotype. So for that the best thing was the golden experiment what Mendel conducted was the test cross. So he named this particular test cross as dihybrid test cross. Yesterday we have seen in the first video class that it was the monohybrid test cross. Now it is called as the test cross conducted for dihybrid cross is called as dihybrid test cross. The test cross which is conducted for monohybrid is called as monohybrid test cross. Now let us see dihybrid test cross now. Dihybrid test cross. Okay, what is dihybrid test cross now? So what is test cross? Test cross is nothing but it is the cross between F1 hybrid and the recessive parent, double recessive parent. That is the definition of the test cross. So what is F1 hybrid now? F1 hybrid cross with recessive parent. So that is capital R, capital Y, small y, cross with small r, small y, small y. So in this experiment, Mendel is going to prove that F1 hybrid is not homozygous, it is heterozygous. So now gametes produced by this are as usual capital R, capital Y, capital R, small r, capital Y, and small r, small y. Here only one gamete because homozygosity, small r, small y. So again checkerboard has to be put now. Checkerboard because it is to be 4 into 1 because only one gamete is present here. 1 is enough here, and 4 gametes are present here. 4 are required here. So male gamete and female gamete, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And here only one gamete, small r, small y. So 1, 2, 3, 4. So you can multiply and write the things inside the squares now or in the rectangles. So capital R, capital Y, small y. Then capital R, small y, small y. Capital R, capital Y. Small r, capital Y, small y. Small r, small y, small y. Okay. What is the first plant available here? Again, depending upon the dominant and recessive nature, we have to decide. Capital R is present and capital Y both are present, so it must be round yellow. So one plant is round yellow. For example, you have right here round yellow. Round yellow, one plant is available. And second one, capital R is present. Due to the presence of capital R, it must be round, but due to the absence of capital Y, both small y are present, it must be round and green. So it must be round green. So it must be second plant. So I can write here one plant. And third plant, here capital R is not there, so it cannot be round, it must be wrinkled. But capital Y is present, it must be yellow. So wrinkled yellow. Wrinkled yellow. So again one plant is available. And the last plant as usual, it must be wrinkled green. So it must be only one. So the dihybrid test cross ratio we can write here 1:1:1:1. So it is the dihybrid test cross ratio 1:1:1:1. But monohybrid test cross ratio is only 1:1. But here different plants are produced here. Because of that, it should be called as 1:1:1:1. So this is called as dihybrid test cross. So totally test cross means it is the cross conducted between the F1 hybrid and the double recessive parent. So this is called as dihybrid cross and test cross. So there is one more cross, that is back cross. Back cross, it is also important. Back cross. So test cross is we know, but the back cross means what? Back cross means, so please remember there must not be any confusion. Test cross is the cross between F1 hybrid and double recessive parent. But back cross is nothing but it is the cross between F1 hybrid and any of the parent. It may be dominant parent or it may be the recessive parent. So back cross is the cross between F1 hybrid and any of the parents. So always in genetics we call all test crosses are back crosses. Please try to understand, all test crosses are back crosses, all back crosses are not test crosses. So again, don't get confused. Back cross is nothing but a cross between F1 hybrid and a dominant F1 hybrid and with any of the parent. Test cross is the cross between again with F1 hybrid and with only the recessive parent. So this is the major difference. So this completes the Mendelism. So Mendel's work is over here. So try to understand, Mendel has conducted a series of experiments only on garden peas. Mendel has conducted experiments up to this. So here the Mendelism completes. So Mendel, after the completion of, after the death of Mendel in 1884, so after when Mendel expired or died, so people got the inspiration and they started thinking of genetics. Now they started thinking, okay, Mendel has done experiments only in garden peas, but what about other plants? Whether the results are same or different, we have to think of that. So then people took the different varieties, different plants, and they started crossing. This is called as Neo-Darwinism. Neo-Mendelism. After the death of Mendel, whatever the genetic work is done, that is called as Neo-Mendelism. And what is the work done during the life of Mendel? What Mendel actually done, that is called as Mendelism. So this is the experiment what I am going to tell you now, that is called as the Neo-Mendelism. After the death of Mendel, some work was taken out. So that is called as Neo-Mendelism. So whatever the results, for example, till today, if you take garden peas and cross, what Mendel has given, those results are true today itself. So, but the same thing when the different plants are taken, what Mendel has told about the genetics or what about the crossing results, those results will not come into true. So now what happens? So here some of the scientists, they took the plant what is known as snapdragon, or it is also called as Indian 4 o'clock plant. So Indian 4 o'clock plant, it is commonly available in Dharwad city and some other places also. So why it is called as 4 o'clock plant? Because the flowers bloom only in the afternoon. After the afternoon or in the evening time they bloom. Because of that it is called as 4 o'clock plant, and in botanical terms it is called as Mirabilis jalapa. Mirabilis jalapa. So this plant was taken and the crossing work was done. So when the crossing work was done by using the Indian 4 o'clock plant or snapdragon, so in Indian 4 o'clock plant there are two colors are available in the plant, two colors. One plant is producing purely red colored flowers and another plant was producing white colored flowers. And the scientists were, after the death of Mendel, the scientists were very much interested. If I take red colored plant and cross with white colored plant, what happens? What would be the result? So here, red colored plant, red flowered plant was crossed with white flowered plant. So red color, capital R, white correspondingly small letter, small r. Okay. But in the Mendel case, when Mendel took monohybrid cross, capital T cross with small t, F1 generation, capital T, small t. And capital T was dominant gene and small t was recessive gene. The F1 plant used to show the character of the dominant parent, that is tall only. But here, capital R is the gamete, small r is the gamete here. After the the hybridization, the F1 hybrid plant or capital R and small r, we are calling it as hybrid and it is called as F1 generation. But in the F1 generation, as per our present knowledge given by Mendel, so it must be red colored. But the things are totally different because the plant is different. So Mendel's laws and rules and the proceedings will not work out here. So this plant was not totally red or totally white. It must be in between. So it was pink colored plant. It was pink. So here what the scientists say that in case of capital R and small r, capital R is a dominant gene, no doubt, but capital R could not suppress the expression of small r. So this is called as incomplete dominance. The capital R is dominant gene, but it could not control the expression of the small r. Because of that, both the genes are expressing here. Capital R is producing red colored pigment, small r is producing white colored pigments. Red and white together form the the pink color. So since both the genes are expressing here, we are calling this one as capital R, the dominant gene, could not successfully suppress the expression of the small r gene, and this is called as the incomplete dominance. Okay, and he comes to parental generation 2. Pink colored plant is taken and self with the pink colored plant only. So here capital R, small r, capital R, small r. Gametes produced here are two types, capital R, small r, capital R, small r. Okay. So if you put the checkerboard here, the results are very interesting. Unlike this one. So this is the checkerboard. Rules are same, male gamete, female gamete, capital R, small r, capital R, small r, again 1, 2, 3, 4. Now multiply and write inside capital R, capital R, capital R, small r, capital R, small r, small r, small r. So here, first plant we take now, first plant is purely red colored because both are capital R, capital R. Last plant, that is the fourth plant, is purely white because small r, due to the absence of capital R, small r is only expressing, so it must be totally white. But the second and third plant, they are not neither red nor white, they are pink color. So here the red, white and pink color plants are available in the ratio 1:2:1. So this is called as the ratio of incomplete dominance. So this is what is known as the genetics class, Mendelism, and I have completed here. But ratios, when you are remembering, first ratio I gave was the ratio of monohybrid cross, that was 3:1. Ratio of monohybrid test cross, 1:1. The ratio of dihybrid cross, that is 9:3:3:1. And the ratio of dihybrid test cross was 1:1:1:1. And the ratio of incomplete dominance is 1:2:1. So don't get confused. Of course, in the next classes we are getting some more ratios in the genetics. So until today we have to remember so many ratios, and as usual, if you have any doubt, you are free to contact me on my phone number 9448821 So I am very happy if you have any queries. That is the first one thing. And next video class I will give something about genetics only. So after the genetics or this one, I have to give something about chromosomes, something. Let us complete one and one. So you seriously go through the video class and also the notes which I have given in my WhatsApp group and also in Google Classroom. Study well and get good knowledge. Have a good luck.